Now we're going to start chapter 11. So if you guys remember, we said that the nervous system is covered over 3 chapters. So this is the second of them. And this discusses the divisions of the nervous system. We already introduced that the nervous system is made out of a CNS, or the central nervous system, and the peripheral. So for the central, that's your brain and spinal cord. And that is really the part of the brain or the part of the nervous system that has the more complicated processing of all of the data that we're receiving throughout the day, different perceptions, movements, decision-making, your personality and your thoughts and so on. The brain is made out of the 2 cerebral hemispheres. So there's a right and a left one. And this here, right here, we're seeing the right only. There's the diencephalon. And that is the portion that tends to be a little bit deeper and in between the 2 cerebral hemispheres. There's, again, a right and a left diencephalon. And then there's the brainstem, the midbrain pons, and the medulla oblongata. And then there's the cerebellum. So your brain, we technically usually think of the brain as just this portion, but all of these together make our brains. The brainstem is what connects the cerebrum of different parts of the brain to the spinal cord. So that continuation downwards would be the spinal cord. Now, the brain and spinal cord are going to give off nerves. Those nerves are known as the peripheral nervous system. Because these structures are kind of important, we have 3 layers that cover them, and these are protective layers. They're known as meninges. We'll talk about them right now. So the meninges, again, are your protective layers of the brain and spinal cord. From superficial to deep, there are 3 layers. So you want to know them in order. Okay, the first one is the dura mater, and dura comes from the word durable. It's the strongest of the 3. It's made out of very dense connective tissue. This is where we've got dural sinuses, and dural sinuses are where the venous blood is going to get out of the brain. So the brain doesn't really have veins like other organs do for the deoxygenated blood. It actually has dural sinuses where these dura layers tend to split and kind of make a tube where the venous blood will be passing through. So they're not real veins. They're again known as dural sinuses. The space above the dura is known as the epidural space. So if you've ever heard of an epidural anesthesia, this is the space where the anesthesia would be injected to. And obviously an epidural anesthesia is not given in the epidural space surrounding the brain. But remember that all of these meningeal coverings surround the brain and the spinal cord. So the epidural injection will be given in the epidural space down in the lumbar area or in the sacral area. The second or the middle layer is known as arachnoid matter, and it comes from the word spider because it actually does look like a spider web. And in the layer or in the space underneath it, known as the subarachnoid space, that there is a fluid that is circulating in there, and that fluid is responsible for bringing in nutrition and bringing in nutrients, getting rid of waste products, and that fluid is known as the cerebrospinal fluid, or CSF. The third of the layers is known as pia mater. That's the deepest. Okay, and that is literally attached to the brain and spinal cord. Like you literally have to peel it off the brain tissue and off of the spinal cord tissue. It has the blood vessels and the nerves. So yes, our spinal cords have do have their own nerves, and it also nourishes the CNS. So here you can see the meninges. So we have these layers of the scalp, and actually scalp stands for skin, and then C stands for the connective tissue, A stands for aponeurosis, L stands for loose connective tissue, and P stands for the periosteum of the skull. And when you remove all of those layers, you are now going to find the 3 layers of the meninges. So we have, again, the outermost one, or the most superficial, and that is the dura. As you can see here, the dura mater tends to split in certain areas, not all around the brain, but in certain parts, it'll split into, to make like these little tunnels, and you would have deoxygenated blood passing through those tunnels, and those tunnels are known as the dural sinuses. You could see in them arachnoid granulation, and when we get to the CSF, we will learn what arachnoid granulation does. Okay, above the dura would be the epidural space, so that little tiny space between the dura mater and the scalp, in the dura mater and the skull, that would be the epidural space. Now for the arachnoid, that is the middle layer, and that is kind of like the that pinkish layer. You could see those spider webs, okay, into the subarachnoid space, and that blue that is filling in the space is your CSF, okay? And then we have the third or the deepest layer, and that is the pia, and that will be this layer right here, and you see that it is directly attached to the substance of the brain and spinal cord. Whenever the brain goes down into a sinus or up into a gyrus, the pia mater goes up and down with the substance of the brain. Okay, pia actually in matter means like the loving mother, so the pia is kind of like hugging the brain, just like a loving mother would do. You can see here the meninges of the spinal cord, because remember these meningeal layers will go all the way down surrounding the spinal cord. So again, you could see the dura mater, that is the outermost, arachnoid in the middle, and then the pia deep. So you want to know these layers in the order from either deep to superficial or superficial to deep. Okay, and right here you can see this transverse section of the spinal cord, and we'll kind of go over the different parts in a later time when we talk about the peripheral nervous system. But for now, I just want you to focus on the meninges that you can see a lot better on this image right here. Now the ventricles and the CSF. So the CSF is produced in 4 ventricles. The ventricles are basically cavities that are found within the brain. Okay, so each cerebral hemisphere has a ventricle, and those are known as lateral ventricles. So you want to think of these ventricles of the brain. These are totally different than the ventricle of the heart. Okay, but they have the same name with the same spelling, but they basically mean cavities that are found within the brain. Again, 2 of them are found in the cerebral hemispheres. These are known as the 2 lateral ventricles. And then there's a 3rd ventricle, and that is found in between the diencephalons. And then there's a 4th ventricle, and that's found in the brainstem. We'll take a look at the image to kind of figure out the different parts. So here you go. So here are the 2 cerebral hemispheres, and these are the cavities in there, and those are known as your lateral ventricles. And then we have the interventricular foramen, which means the opening in between the 2 ventricles so that the CSF fluid can drain down or pass down to the 3rd ventricle. So the 3rd ventricle is really found in this space. It's a very tiny space found in between the 2 cerebral hemispheres, and we call that the 3rd ventricle. And then the CSF will pass through a duct of water, hence the name cerebral aqueduct. It's known as either the cerebral aqueduct of Sylvius or the cerebral aqueduct of the midbrain. Sylvius is just the scientist that discovered it. And then there's a 4th ventricle, and the 4th ventricle is found over kind of at the level of the brainstem. Here again, you could see it in a lateral view. So this is what a lateral ventricle looks like. It kind of looks like a C-shaped cavity. And then you have, again, the CSF will flow into the interventricular foramen, down to the 3rd ventricle, and then through the cerebral aqueduct of the midbrain down to the 4th ventricle that kind of looks like a triangle. And then the CSF would go through a little tiny canal known as the central canal of the spinal cord in order to go down all the way through the spinal cord. Now, where is CSF made? Or how is it made? And how is it drained? Okay, CSF is a very constant fluid. So the CSF that we were born with is definitely not the same that we have right now. Okay, we are constantly getting rid of the old CSF because, again, it washes away some waste products, and we are constantly making fresh CSF. So how do we make fresh CSF? It is secreted by the choroid plexuses, and the choroid plexuses are made by the ependymal cells. So if you remember from the previous chapter, we said that the ventricles in the brain are lined with ependymal cells. These ependymal cells, we said at the time, make CSF. Okay, so this is just repeating what we've already learned about the different cells that are found, the neuroglial cells that are found in the CNS. Okay, it'll selectively transfer substances from blood in order to make CSF. So the consistency of the different chemicals in the CSF is not the same as it is in blood, okay? So CSF would have like a higher glucose level because your brain would need it. It has different ion levels, sodium and potassium levels. So these ependymal cells are making sure that we're changing the consistency of the blood and plasma before it pumps it into the subarachnoid space. You'll find that it has lots of nutrition, lots of protection that the neurons are going to need. It keeps the ionic concentration stable within the CNS. So when we talk about how an action potential works, we talk about lots of sodium and potassium ions that are needed. So we want to make sure that these ion concentrations are within normal ranges. That CSF is going to circulate in the ventricles, kind of as we mentioned, to the third ventricle, then down through the cerebral aqueduct to the fourth ventricle, down the central canal of the spinal cord, and then it will be absorbed by the arachnoid granulations. So remember we talked about these arachnoid granulations that are in the dural sinuses. These are going to absorb what is now old CSF, okay? Taking with it the waste products. The amount of CSF that we have at any time is about 140 milliliters. And if you switch milliliters to fluid ounces, it's kind of like between 4 and 5 fluid ounces. So it's about half a cup, which is not much, but that CSF is extremely important to our brains. So a little clinical application, sometimes the CSF should have a normal pressure. Okay, so we make some and reabsorb some. The amount that we make should be equal to the amount that is being reabsorbed through the arachnoid granulations. So we have a constant pressure of CSF. But in some cases, there is either overproduction or there is a blockage in the drainage system. Okay, and that could lead to cerebrospinal fluid increase in pressure. Okay, and if that happens in a kid where, remember, the different bones of the skull are not completely ossified. So we still had the anterior and posterior fontanelles. So the brain is able to swell up and it will pull those bones away from each other. And you can see it in this condition right here. So this is pre-surgery. Okay, that is known as hydrocephalus. Hydro literally translates into water on the brain. Okay, they can be treated and you want to just drain all of that extra fluid. So a shunt would be introduced and that would drain the excess water or the excess CSF from the brain into the peritoneal cavity in the abdomen. Okay, so that shunt would obviously the kid is growing, so that shunt would have to be changed constantly in order to get taller ones so that the drainage is not blocked or does not drain into another area besides the abdomen. Okay, we could also do a spinal tap or a lumbar puncture where we insert a needle into the lumbar area into the subarachnoid space and drain some of the CSF. That would have to be done very slowly though, because you want a gradual decrease in pressure, not a sudden decrease in pressure. The things that could interfere with the circulation or block the circulation could be maybe a tumor or blood clot, an infection. Okay, sometimes individuals would have a what is known as an ependoma, and that would be a tumor of the ependymal cells. So an ependymoma is a tumor of the ependymal cells. Remember, these are the cells that are secreting the CSF. So if you have a tumor of them, then these individuals will be making CSF at a very faster rate than the drainage. Another clinical application is TBIs, or traumatic brain injuries. And those are, where the brain is exposed to mechanical force. It could be maybe a car accident, a sports injury, a fall. The TBI could range all the way from a very mild concussion where, the patient comes in, they've got this mild headache, and it'll just go away in a couple of days. Okay, but concussions, though, can be very dangerous and could lead to long-lasting symptoms. We also have CTE, that is chronic traumatic encephalopathy, and that is usually sports-related, especially if they're mild repetitive TBIs, where, for example, like in boxing, these results from multiple small injuries that happen over time. The symptoms usually begin years later. So you have, these athletes that maybe are into boxing and then they get multiple mild concussions. When they stop playing, maybe 20, 30, 40 years later, that's when they start experiencing symptoms, and they would have long effects on memory and on behavior. There's blast-related brain injury, and that is a very severe form of traumatic brain injury. And that could be, for example, in combat situations where the patient is exposed to these explosions, and that could lead to cognitive decline even years later after the injury. Now we'll talk first about more detail about the brain and the different brain centers and so on. The brain has these centers for different senses like speech, hearing, vision, and so on. But it's not just feeling things. It is having the ability to analyze what we felt, to interpret these things. How do we change our behavior, our thoughts, our judgments? So the brain is has a more sophisticated function as opposed to just feeling or hearing and so on. So it has these higher mental functions like memory, reasoning, judgment, decision making, and so on. Although it is still, responsible for coordinating what are kind of like more primitive things like digestion and the smooth muscle movements and so on. The major portion of the brain are made, as we mentioned already, so this might look familiar to you folks, the cerebrum, the diencephalon, the cerebellum, and the brainstem. So how did we develop these parts? So a little bit about embryology, which is the study of the science of the embryos. How did we develop? How did our brain start to develop? Generally speaking, our whole body really was just at one time a flat layer of cells, and then that layer of cells became rolled over into a cylinder. Okay, now we're in a tube, and each part of that tube is going to make different things. So the part that will develop into the CNS is known as the neural tube. The brain is going to be formed out of these three little vesicles, the forebrain or the prosencephalon, and that will develop into the cerebrum and the diencephalon. The mesencephalon, also known as the midbrain, that will develop into the midbrain. And the hindbrain or the rhomboencephalon, and that is going to develop into the pons, medulla oblongata, and the cerebellum. Now, what does it look like now that we are born and we have a fully developed brain? So the cerebral hemispheres, again, we have the right and the left, but they are separated in the middle by a layer of dura mater, and that layer of dura mater is known as falx cerebri. So that layer of dura mater would be in this fissure between the right and the left ventricles. That fissure is known as a longitudinal fissure. There will be a sheet of dura mater separating the two cerebral hemispheres from each other. And again, that sheet of dura mater is known as the falx cerebri. But it is not a total separation. So there are some fibers, it'll separate from the top only. It'll just want to go back. So that the falx cerebri would be all the way up until here. But there are fibers, so these white, kind of like a C-shaped fibers, connect the right and the left cerebral hemispheres to each other. And because it is a big body, we call it the corpus callosum. So these corpus callosal cells connect the two cerebral hemispheres. Now we have ridges or convolutions, and those are known as the gyri or gyrus, which is just one. It can also be pronounced gyrus. We also have the grooves. The shallow grooves are known as sulci. Sulcus would be one. Okay. One of the most important sulci you want to remember is the central sulcus, which is this sulcus right here. And that separates the frontal lobe from the parietal lobe. The deeper grooves are known as fissures. And the two fissures that we want to be familiar with are one that we already talked about, and that is the longitudinal one that separates the two cerebral hemispheres. Another one that you want to be familiar with is this transverse fissure right here, and that separates the cerebellum on the bottom from the cerebrum on top. Actually, there's a third one. You might want to add it on here, and that is the lateral sulcus. The lateral sulcus separates the temporal lobe below from both the frontal and the parietal lobe above. These are the 5 lobes of the cerebral hemisphere. Okay, if you were studying this, I would say maybe when I was studying this about 25 years ago, there were only 4 lobes. Okay, not that we developed a new lobe, it was just there, and we never knew that it was a separate lobe. So now we know that we have 5 lobes in the cerebral hemisphere. Remember, 5 on the right, 5 on the left. 4 of them are named according to the bones that are found on top of them. So there is an orange frontal lobe. Obviously, we're not color-coded, but it just helps to look at them. The frontal lobe is in the front, and it lies underneath the frontal bone. It is in front of the central sulcus. Behind the central sulcus, underneath the parietal bone, is the parietal lobe. All the way in the back, underneath the occipital bone, is the occipital lobe. And then on the side, underneath the temporal bone, is where you can find the temporal lobe. The insula you cannot see it in this image. You would have to remove the temporal lobe in order to see it. So the insula is named that because of how it is insulated. It is very deep in the structure. Again, you would have to remove this temporal lobe in order to find the insula. If you look at the structure of the cerebrum, it's made out of a cerebral cortex. That cerebral cortex is a very thin layer of gray matter. So what do we mean by gray? We mean that those are the parts of the nerves that are not myelinated. So they're collections of cell bodies and dendrites. That gray matter has non-myelinated structures. That is the most outermost layer of the cerebrum, and about 75% of our cell bodies are going to be found in the cerebral cortex. Underneath the gray matter is a white matter. White comes from the fact that these are myelinated axons. Remember, myelination are made out of basically the oligodendrocytes, where that would make your myelin sheath in the CNS. They're basically fat or lipids, and that's why they get that whitish color. And we call that area the white matter. Okay, so it has bundles of myelinated axons. These myelinated axons are going to connect different parts of the body, the brain, to each other. Now, what is the function of the cerebral cortex? As I mentioned before, it is for higher mental functions. So, yes, I am going to hear with my temporal lobe, but I'm also going to be able to understand what I heard, interpret it into, analyze that data. Maybe it'll bring back memories. I can form memories, form new ones. It will maybe make me feel happy or make me feel sad. So all of these analytical and processing details that you can do is done by the cerebral cortex. You could even retrieve information, so memory, okay, reasoning, decision making, okay, your intelligence and personality, all of these would be thanks to the cerebral cortex. We'll talk about the functional areas of the cortex. So which parts of the cortex do what? Well, the cortex can be divided, just like any other part of the nervous system, into a sensory area, motor area, and then there's also an association area. So we'll talk first about the sensory area. And the sensory areas are obviously the areas that are going to gather sensory information. And I'm not going to use this image. This has all of them put in there. So we'll do them one at a time. So we'll talk about the sensory areas of the cortex. First off, I'm actually going to leave this cutaneous sensory area to the last. I want to talk first about the sensory speech area or Wernicke's area. And that is that green area right here. And you can see here it's taking part of the parietal and part of the temporal lobe. This is found usually on your dominant hemisphere. Most of us have a dominant hemisphere on the left side. So we will have this Wernicke's area well developed in the left hemisphere. It is responsible for understanding and formulating language, which means that, let's say, for example, I'm able to understand and speak English and Arabic. So my Wernicke's area would have the software for both of those languages. I can still hear other languages using my temporal lobe. Okay, that's the auditory area, but I do not understand it. I cannot speak it. I cannot formulate a sentence using those languages, which means again that I do not have that software. So in order to learn a language, in order to learn how to understand and speak a language, you would need the software in the Wernicke's area. That is known as your sensory speech area. Okay, because we'll learn that there's a motor speech area. So know that the sensory speech area found in the temporal and parietal lobe is responsible for understanding and formulating language. Okay, if you lose that, okay, you can still hear it. You can still hear languages that you are familiar, that you know, but you are going to lose the understanding and the ability to formulate a sentence. Okay, unfortunately, these individuals are not aware that they have this problem, so they will be talking, thinking that they are communicating their thoughts, not realizing that their sentences make no sense. Okay, so these, again, patients are not aware that they do have a problem. Now for the visual area, that's found in the occipital cortex and that all the way at the tip, and that interprets vision. So the ability for me to see something and look at something and actually see it is due to the visual area in the occipital cortex. The auditory area is in the temporal lobe, and that helps me interpret hearing. So listening to music and so on. So just interpret the hearing. When I can recognize that this is a song. Going into more detail about the song is of the association areas that we'll talk about in 1 minute. There's a sensory area for taste, and that is near the base of the central sulcus. So remember the central sulcus, all the way deep here, it's not really shown in this image, but near the base of that central sulcus, it includes parts of the insula. That's the sensory area for taste. The sensory area for smell, that as well is not shown in this image. That's very deep within the temporal lobe. Okay, that's where you can find them. Now for the cutaneous sensory area that we mentioned we'll talk about last, that is found right here in this gyrus in the parietal lobe. It's known as the post-central gyrus because it's found right behind the central sulcus. This interprets, that yellow gyrus interprets sensations coming from the skin. So how is it, how is our body represented on the that sensory cortex. So this right here is just one lobe. Okay, that's the longitudinal fissure. This would have your right lobe on the opposite side. We're looking here at the left side. And you can see here that this is the medial part of the cerebral cortex, and that is the lateral portion of the parietal cortex. When you look at how the different parts of the body are represented on the parietal lobe, you can see here that it looks like we're kind of hanging from the feet and the genitals on the medial side, and the rest of the parts of the body are represented on the lateral side. So feet, lower limbs, and genitals on the medial side, and the rest of the body on the lateral parietal cortex. This is showing you sensory areas. This is showing you motor areas, which we'll see down the road. The association areas of the cortex. So our association areas, these are the areas that are going to take information from different sensory areas and integrate them together in order for us to get a better understanding of what's going on. These are not considered motor. They're not considered sensory. They, again, integrate sensory and motor together. They would integrate more than one sensory area. So, for example, in order to provide a memory, reasoning, or judgment, or emotions. So, for instance, if you're listening to the listening is right up here. Listening to the music in the auditory area, and you're looking at a performance using the occipital lobe, you want somewhere in your body to be able to integrate both of those information together to know that to give you that full experience of what you are watching and hearing. Okay, so those, and then, the feelings that are going to be produced because of that experience. So all of those are going to be, again, done in the association areas of the cortex. So let's take a look at the different association areas. Each lobe has its own. So the frontal lobe association areas are responsible for concentrating and planning, complex problem solving, emotional behavior, and judging consequences of behavior. So the frontal lobe, like as you're trying a test to answer a question, and you kind of have your hand on your forehead, trying to think, concentrate here. Okay, so that is your concentrating area, the frontal lobe association area. You're planning, planning like a trip. All of that information is being processed through the frontal lobe. The emotional behavior, so how do I respond maybe to somebody? Do I actually do that response, or do I change it a little bit because I might get in, the consequences of my behavior are telling me maybe I should do it, maybe I should not. So all of these different thoughts are processed through the frontal lobe association area. The parietal lobe association area helps in understanding speech and to choose the words to express thoughts and feelings. So when you're trying to express your thoughts and you're trying to kind of dig deep into the right words to use, that would be your parietal lobe association area working. The temporal association area, that is to understand complex sensory experiences, like understanding speech and reading, and not just like understanding what is being said, but maybe going deeper into that, like understanding what might be behind that speech or behind what I'm reading. It is also where we store memories of visual scenes. Okay, so the different scenery, the different music, and so on. The occipital association area, that's where we analyze and combine visual images with other sensory experiences. So again, watching a movie, you're using your occipital lobe, but analyzing the movie, thinking about, did this bring back memories or trigger things in me? Does it motivate me to do, to be better, and so on? All of these different deeper analyzing of that visual experience would be due to the occipital association area. Insula is where we translate sensory information into proper emotional responses. So your insula, let's say, I'll just give you an example here, to kind of compare what that means. So let us say you're watching TV and then all of a sudden there is a lion roaring. Okay, so you're hearing the lion roaring using your temporal lobe. You can see it using the visual lobe that it's on the TV. What would your response be? Okay, so your response would be, this is on TV, so there's nothing to be afraid of. I don't need to get up and run. Okay, so we've translated all of that sensory information into a proper emotional response. Let us say we hear a lion roaring, but now we see that we are actually in the jungle, okay? So your proper emotional response in that situation would obviously be very different as opposed to sitting in your living room watching it on TV. Okay, so the insula is responsible for translating that sensory information into what would the proper emotional behavior be given the situation. Now for the motor areas of the cortex, there are three that we want to know. There is the primary motor area, and that's that pink, also known as the precentral gyrus. Again, the gyrus found in front of the central sulcus. Primary motor area, it is responsible for controlling the voluntary muscles of the whole body. Okay, these fibers would obviously, in order to reach whether the upper limb or lower limb, would have to pass through the brainstem and the spinal cord. Broca's area, and that is the motor speech area. So remember we talked about sensory speech area. This is your motor speech area, Broca's area. It's found in the frontal lobe on the dominant hemisphere, and that controls the voluntary muscles that we use for speech. So we use the tongue muscles, we use the laryngeal muscles, and all of these different muscles in order to give us these different letters and so on. All of those muscles are controlled through this Broca's area. The frontal eye field, and the frontal eye field, I don't see it on here, but the frontal eye field is right above Broca's area, and that it controls all of the voluntary movements of the eye and the eyelids. In Broca's area, because we want to talk about motor aphasia, which is the inability to speak, that would be aphasia due to a motor lesion. So motor aphasia is where the individuals have damage to this Broca's area. It could either be due to a stroke, or a tumor. So these individuals can understand speech. They are able to formulate, so they can put the sentence together, but they cannot actually say it. They have lost the ability to control their voluntary muscles of the tongue and so on. So they have lost the ability to speak. They know what is going on. So these patients are extremely frustrated because they do have their thoughts together, but they cannot express themselves using speech, which is very different from when we talked about sensory aphasia, where, again, they are unaware of what they're doing or what they're saying. Now for the motor area, again, how are our voluntary muscles represented on the precentral gyrus? It's just the same as sensory. So again, we are kind of the feet are on the medial side of the cortex, while everything else is on the lateral side of the cortex. A couple of things that I want to get your attention here, or direct your attention to, is you can see here that our hands and the face have a very big area of representation for all of the facial expressions, for all of the movements that we do, especially the fine movements of the hand. You can even see that same representation kind of applies when it comes to sensory areas, because again, this is where we do a lot of sensing using the hands and the face. These are the functions of the cerebral lobes in a table format. Okay, so, some students prefer it this way to kind of, now that we've learned everything, let's put it in a table. So there you go, folks. It's from the textbook. And at this point, I am going to stop, and I will see you folks in part 2.